Thank you everyone for joining us today for PPMD's webinar with Edgewise Therapeutics. I was going to be talking about their investigational product EDG-5506 for both Duchenne and Becker muscular dystrophy. I'm Eric Camino, I'm PPMD's Vice President of Research and Clinical Innovation. Joined today by a few team members from Edgewise Therapeutics. We have Alan Russell, who is the Chief Scientific Officer and co-founder of Edgewise Therapeutics. Joanne Donovan, who is the Chief Medical Officer at Edgewise Therapeutics, and Abby Bronson, who is the VP of Patient Advocacy and External Innovation. As we get into the session today, if you have any questions, please submit them to the chat function. We will try to get to all of the questions during the open Q&A at the end of the presentation. We're also recording this webinar, so if you have to step away or if you have friends or family who you think this would be valuable to, please let them know. We will host this on PPMD's website following the talk within the next week to review then. So with that, I'm going to hand it over to Abby Bronson, who's going to get us started for today's talk.
Good afternoon or good morning, everybody, wherever you happen to be. I'm so pleased. We at Edgewise are really pleased to be here, and we thank PPMD for organizing this. And I'm just going to kick it off and just sort of do a brief introduction about our product candidate, and about Edgewise. So you can go to the next slide. So this is just our forward-looking statement. I think you've probably seen a lot of these. Edgewise is a publicly traded company. We're all employees of Edgewise, and EDG-5506 is an investigational agent, and it's not approved in any territory. You can hear, I think some people call it EDG-5506, some people call it EDG-5506, but both mean the same thing. So next slide, Alan. So just a little bit about Edgewise. It was launched in 2017 in Boulder, Colorado. And as it said on the first slide, Alan was one of our co-founders who really was the mastermind behind this novel way of looking at muscle breakdown. Our vision is to improve the lives of patients and families suffering from rare muscle disorders. And as we've said, our approach is novel. So this is a first in class, no one else is doing this. And it's potentially complementary to some of the current approaches. And this is done by, and Alan's going to talk a lot about this, directly modulating or targeting a key muscle modulator. So he is going to give you a deep dive into our approach and our mechanism of action, which is just a fancy way of saying how our investigational drug is intended to work. So the lead program what we're talking about today is EDG-5506. It's focused on Becker and Duchenne. We've successfully initiated a phase 1 and to look at safety in dystrophic tissue. And Joanne is going to take a deep dive into that. And she's also going to discuss our ongoing trials and our upcoming trials in Duchenne. And you can go to the next slide, Alan. So before we get to Alan and Joanne, I want to take a moment just to talk a little bit about how we approach patient advocacy at Edgewise. And truly, patients are at the core of everything to do, and we are better when we listen to you. So I like to think of what we do as being divided into two parts. And the first part is really listening to you and gaining your perceptions, your thoughts. What's it like to live with Duchenne, either as an individual or a caregiver? And we use these insights to help make our trials easier, try to lessen the trial burden where we can, and also to establish something that we call the therapeutic context. And what I mean by the therapeutic context is that, you know, as all sponsors do, all companies do, we do trials, we collect a lot of data on safety and efficacy, and ultimately all that data is used by the regulators to make a benefit risk decision. But without knowing what are the symptoms, what's it feel like to have this disease, what are the unmet needs, they can't make, it's harder to make a benefit risk decision. And that's what we mean by the therapeutic context, really understanding the burden of disease and the unmet needs. You might have heard of the acronym PFDD, or Patient Focused Drug Development. And this is, the therapeutic context is all about this. It's all about incorporating the patient voice into the benefit risk decision. So the second part, and basically probably the last 3 bullets of this slide, is really working collaboratively with the community to help accelerate drug development and increase awareness of the dystrophinopathies and some of the rare diseases that we work in, And this can happen in all different ways. We present at very local events and participate in all those very small activities. We also participate in, larger collaborations and global consortiums, working on MRI, working on novel trial designs to help accelerate drug development in Duchenne, which is ultimately our goal. Okay, Alan, you can go to the next slide. So what's our approach at a very high level? It's really very simple, and that is to protect susceptible muscle fibers from damage during contraction. And what's been reported in the literature, and then we confirm this, and Alan can go over this a little bit more, is that when muscle fibers break down, some of them are more susceptible to damage, meaning that they get damaged more easily. So we've designed our investigational therapy, EDG5506, to protect these susceptible muscle fibers. And it doesn't matter what mutation you have. This will work for or potentially will work for everybody, goes for everyone. We looked in diseased animal models, and EDG5506 was found to protect these susceptible muscle fibers and prevent long-term damage. So that was sort of the proof that this hypothesis was something to follow up on. So now I'm going to turn it over to Alan, who's going to take you even deeper into some of the science.
Good morning, afternoon. So what I'm going to do is actually show you dystrophin in action. So rather than a static picture, that's the kind of thing we have on the left-hand side, I'm going to show you what the protein does in contracting muscle. So what you're looking at the right-hand side is muscle contracting, and the dystrophin complex, as it were, are those green blobs. And what they're doing is spanning the membrane from the inside to the outside of the muscle and connecting these fibers up. And they can connect fibers whether one is contracting or all of them are contracting. So you can see this pink muscle fiber in the middle is contracting and the other ones are not. That's a normal physiology. And the dystrophin is supporting that contraction. Now in Duchenne, for instance, those dystrophin elements are not there. And the body tries to keep up with this by upregulating different elements, but they don't do a great job. And what it really means is these poor fibers are contracting on their own. They don't have the support from the surrounding fibers, and that leads to stress. You often hear about CK, and I'll get back to that in a little bit. It's really because they get membrane stress, and that causes this local stress in the muscle fibers, and that really builds up with time in these injured fibers, and eventually that leads to stress building up, activating enzymes which chop up the fiber. Now that happens in you and me, even if we don't have Becker or Duchenne, when we exercise, right? So if we do weights or something, that's how we adapt our muscles to improve our strength, by injuring our fibers in a controlled way. But of course, if you have Duchenne or Becker, the barrier for injury is much, much lower. So what we're going to hope to do is stabilize these fibers so that when they are alone in the world and don't have their supporting friends, they can still contract without injuring themselves. So we're not replacing or modifying dystrophin in any way. What we're doing is stabilizing the fiber with this compound that Abby just described, 5506. This is just I'll just skip over this one. That was just in case the video didn't work. So what you're looking at now is the same thing, but actually live muscle. So this is muscle from a mouse. It's actually a finger muscle. You can see right through it through a microscope. On the left-hand side is a non-treated muscle. Okay, so this is a MDX mouse. And as you can see around here, you see these kind of breakage events. This is exactly the same as in the little video that I just showed you. So the individual fibers are getting stressed, and then the enzymes are snipping them up. And what you're seeing is these chopped up fibers now crushing into themselves to essentially make a ball of protein, which is then digested and disposed of to allow you to grow a new fiber. Now that happens all the time in our bodies, also happens in Becker and Duchenne, but the inflammation that causes eventually catches up with you. And that's what causes the fibrosis and the fat that leads to the functional problems. In the right-hand side here, we've treated this muscle for 1 hour with 5506. What we've done is we've modulated how the muscle contracts without actually affecting the overall function of the muscle, But now when the muscle contracts, the membrane does not get stressed and you don't get these injury events. So we've stabilized those individual fibers so that when they contract, they don't pick up the injury. So of course, if you could do this in the muscles of an individual with Becker or Duchenne, they should be able to be active, but now that's not going to turn into injury that causes the inflammation and the fibrosis. Now, part of that injury, as I mentioned, is leak of proteins into circulation. And many of you are probably aware of creatine kinase, or CK. It's one of the earliest tests that you tend to get prior to diagnosis of one of these conditions. There's other proteins that also leak out of injured membranes, and that includes things like myoglobin. We'll talk about fast troponin, and then there's lots of other things as well. And the reason I talk about fast troponin is we protect the most susceptible fibers, the fast ones. They make up about 50% of your muscle, and this protein is specific for those fibers. So if you like, this is the most sensitive biomarker for us to know whether our compound is doing what we say it's doing. You can see that both of these proteins are elevated if you have Becker or Duchenne. So this is just a variety of people from a cross-section of ages, plasma blood samples, and we're comparing control individuals to either Becker or Duchenne, and you can see Duchenne have high levels of CK, Becker in the middle, and the controls low. And that's also the case for troponin. So Duchenne is high, Becker is in the middle, and the controls are low, and these two levels actually match each other. So if you're high for CK, generally you're high for troponin. I'm going to show you just one additional kind of preclinical data result. We've done a lot of studies in mice, but this is actually in golden retrievers with muscular dystrophy. So these are large dogs, and as such, they get fairly severe disease. And what we did is we did a short dosing study in dogs that had kind of had their disease stabilized. So they kind of reached the adult phase of the disease, if you like. So these dogs are weak and they have low muscle tone. And we looked at two things during two separate studies. One was how much CK was in the plasma of these dogs. And you can see when we dosed the dogs with 5506, the CK level went down by about 50%, so kind of halved. While they were doing this study, the text reported that the dogs appeared to be more active. So we actually went to Amazon.com essentially and bought a lot of activity monitors for your pet. They're called FitBark devices. You can measure activity. And we're really pleased to see in a separate study, when we placed the dogs onto 5506, their activity actually increased. So you've changed this relationship between activity and injury. I'm really hopeful that what we'll essentially be able to achieve in the clinic is a remodeling of muscle in the background of both Becker and Duchenne. And at this point, what I'll do is I'll turn it over to Joanne, and she'll describe why we see Becker and Duchenne as really a kind of continuance and what kind of data that we have in the clinic.
Great. Thanks, Alan. So I'd like to thank everybody for listening today. And as Alan said, we are looking and doing studies in both Becker and Duchenne muscular dystrophy. Now, why is that? And why, if you have a family member affected by Duchenne, why do you care about Becker and vice versa? And so you know that both of them are a disease where there is decreased dystrophin levels. And compared with the dystrophin levels in unaffected people, you know that in Duchenne, dystrophin is essentially absent. And that causes the kind of damage that you saw in the muscles, that in the mouse muscles that Alan was showing you. With Becker, the dystrophin protein is greatly decreased, or it doesn't work right. So it's not able to function the same way those green bars function and give support to the muscle fibers. So in both Becker and Duchenne, what happens is that the muscle gets damaged with contraction. So everyday activity contracts the muscle or damages the muscle. So what we are doing is looking at this as a continuum. And in fact, you've kind of heard this before, because companies that are developing like a microdystrophin therapy or exon skipping therapy, they are talking about converting somebody who looks like somebody with Duchenne and providing more dystrophin, make them more like Becker. So it's basically a spectrum of disease. And so that's why we are looking at both to gain knowledge, to understand the drug better, to understand better how it can work in people. So on the next slide, we started, as Alan said, we started in people. And we started, as many drugs do, we started in unaffected adults. And we also enrolled in this phase 1 study, we enrolled a number of patients with Becker muscular dystrophy. And in a phase 1 study, the first time that a drug is in people, there are a number of important things to figure out. And one of the most important is safety. What do we know about side effects with the drug? And in this study, we dosed people for up to 2 weeks, and we saw that EDG 5506 was well tolerated. The most common side effects were a feeling of dizziness that happened initially and went away, and a feeling of sleepiness. And actually in clinical trials, we're now dosing at night. That tends to happen in the first few days and goes away. And those are the 2 main things that we've seen. Now, how do we give it? It is an oral tablet that can be taken once a day, and it is absorbed either with or without food. So it's pretty convenient to take. It's not an IV drug. And one very important question in developing drugs in general is, does the drug get to where it's supposed to be? So does it get to muscle in this case? And what we found was that EDG 5506 is highly concentrated in muscle compared to in the bloodstream. In fact, there's 100 times more in muscle than there is in the bloodstream. So that tells us that it's getting to where it needs to be to protect the muscle. And in fact, the levels that were reached were the kind of levels that were reached in mice and in dogs where the drug showed a good effect, some of which Alan showed you. So we know the drug is getting to where it needs to be, which is very encouraging. So what we did was we enrolled those patients with Becker in a longer study, an open label extension, like you frequently have after a clinical trial. And what we looked at was whether or not these biomarkers that Alan was telling you about that reflect damage to muscle were reduced. And we saw that. We actually saw that creatine kinase or CK was reduced and stayed reduced over months. We now have data up to 6 months where it's 40% decreased. And this very specific marker that tells us whether or not the susceptible muscle fibers are being damaged. And that was down almost 3/4. And if I showed you the individual patients here, you'd see that they're all going down almost in parallel. Everybody sees their levels drop greatly. So that's very encouraging. And we're going to continue to look at these folks. There's a dozen folks with Becker that are in the study, and we'll be continuing to look at those and functional measures as well as we move on. Now, this gave us the foundation for additional clinical trials. The first one here, the ARCH study, is the one that I just showed you briefly data from. There is also a study in ambulatory adolescent and adult males with Becker, boys that are 12 and over, and that's called the Canyon study. That is a 1-year study It is enrolling now at a half a dozen sites across the United States and is going to be looking at the placebo-controlled trial. It's going to be looking at functional measures, biomarkers, and safety over the 1-year period, and that will be followed by an open label extension. I'm going to take a deeper dive into the initial study in Duchenne that we're planning on starting by the end of this year, starting imminently, and that's called the LYNX study. That is going to be done in the United States. And on the next slide, gives you a little bit more detail. So this is a 1-year study. It's divided into 2 parts. And but the overall time that boys would participate is a year. The major goal of it is to study the safety of EDG 5506 and to identify doses that would be have the potential to best reduce muscle damage. What's really exciting about the studies that we've done so far is that we see that the drug reduces damage or reduces the biomarkers of damage to the muscle right away. So we are able to choose doses to be able to make sure that we've got doses that are reducing those biomarkers. So we'll look at that in this study to then plan the next study. It will include boys age 4 to 9 up to their 10th birthday who have a diagnosis, a genetic diagnosis of Duchenne. They will need to be ambulatory and meet certain functional criteria, with, for example, the time function tests, things like getting up off the floor in a certain period of time. And that's something the investigator would talk more with you about. They need to be on a stable dose of corticosteroids. What that means is they would need to be on them for 6 months. And the reason that we do that is because we know that corticosteroids changes the muscle, that changes the speed and the performance measures that we're looking at. It even can change CK. So we want them to be on a stable dose so we can understand whether EDG 5506 is changing those biomarkers. That's why we want them to be on a stable dose of at least 6 months of corticosteroids. We'll be enrolling in up to about 12 sites across the United States. And some of those sites will be up very soon. And usually, as you know, it takes a while to get all of the sites up and enrolling. So this will be a rolling process. And we'll continue to inform the community of the sites that are open. To that end, we post on clinicaltrials.gov, and that tells you the sites that are able to enroll boys and as they come up to speed, we update clinicaltrials.gov, and we'll also be reaching out to the community through advocacy to let them know when sites are open. So in the next slide is more about the design of the study. Because we see these biomarkers change quickly, we can look for 3 months at those. So it's a 3-month placebo-controlled period. It's 2 to 1 in the treatment period, 2 active to 1 placebo. And that's followed by a 9-month open label period. And that is all of the boys would be on drug for that. And we would anticipate then extending that as well. But right now, when boys or when their families sign informed consent, they would be signing it for 12 months. So we try to minimize the placebo, certainly, as we do in all trials. So during the 12-month period, there are about 10 site visits, and that includes screening, the initial visit where People sign informed consent, learn about the study, and do certain screening tests to identify whether they're eligible, go over their genetic report, things like that. What we have done is also to incorporate measures that would, to be able to follow people at home, things like telephone calls, and I'll show you a little bit about blood tests as well on the next slide. At these visits, what will happen are blood tests for safety, functional assessments such as hand grip, how tightly, how strong the muscles are, North Star ambulatory assessment, which is 17 common measures, things like climbing up a step, climbing down a step, getting up off the floor, walking or running 10 meters. Also measures like climbing 4 stairs and walking for 100 meters and how long that takes. We'll be doing safety monitoring on the heart, such as EKG and cardiac echo. That's non-invasive, no needles, and a DEXA scan, which looks at the bone density, looks at the muscle mass, and that is a kind of an X-ray that has very minimal radiation. And what it does, and there's no poking, the boys just lie down for that test. And we'll also be doing breathing tests at the beginning and periodically. We'll be doing some measures at home. We are using this device called a Tasso device to collect samples for biomarkers, to measure the amount of drug in the blood. And it looks big. It's about 4 inches long, 2 inches wide. And what it is you put it on the upper arm and push the button, and there is a very narrow gauge needle. And since the upper arm, it's designed to use the upper arm because the upper arm is not terribly sensitive like your finger sticks. Finger sticks hurt more than this. I've done it. It's pretty minimal. And that reduces travel to the study sites for routine blood monitoring. We are also going to be using activity monitoring. Alan showed you that in some of the animal studies, we saw an improvement in activities, and this is something that is being used in multiple studies to understand how the boys' daily activity varies. Does the drug have an effect on that? And it's kind of like an Apple Watch worn on the ankle and wrist. And that will be used at certain points during the study to look at how active the boys are, how fast they run in everyday life, because those measures have been shown to correlate with other measures and give a sense of how the drug is working. And lastly, as we look forward, these are our first 2 studies. But how do we make sure that we're developing the drug with the whole community in mind? So we're starting with 4 to 9-year-old boys, but I can assure you we're also talking about how do we look to younger boys? How do we look to older boys and men with Duchenne, with different levels of ambulation, with non-ambulatory population? Right now, we are in the US. Our studies in the future will certainly go into other countries. We are incorporating, as I mentioned, things that are home-based measures to minimize the trial burden. And we are always talking to experts about how new changes in Duchenne and the care will change potentially this trial or future trials. And really, as we are thinking about it, well, we're already in many trials are already looking at combination therapy with a new drug on top of steroids. We're also including boys in the study who are on exon skipping therapy, because we think that the drug can be used potentially either by itself as if with steroids, or with other drugs that are dystrophin targeted, whether that be exon skipping or potentially in the future gene therapy. Because again, with all of these drugs, there's still ongoing damage to the muscle. So if we can stabilize that, if we can protect the muscle, that hopefully will translate into improved function long-term in these boys. So we are, as I've told you, not just in the way we design the trial, but how do we execute the trial to make sure that we're reducing trial burden. We've been listening to the community. We've had meetings, multiple meetings with advocacy groups to get their input on the design of the trial. We know that travel is an important burden, certainly to take the time. And so we are providing a service that provides concierge services to support travel, to minimize upfront expenses, prepaid, and just provide resources to ease the burden, to ease costs, things like if for other children in the family, to support those costs of either bringing them or childcare. And as I mentioned, also at-home elements of the study to reduce frequency of travel. So we can't eliminate that altogether, but we are trying. So to summarize, EDG 5506 is being developed for Duchenne and Becker muscular dystrophy, taken orally, intended to preserve and to improve function in Becker and Duchenne patients with any mutation. The goal is to prevent damage to muscle by protecting the most susceptible muscle fibers. The potential is to be used as a single agent or in combination with other therapeutic approaches for dystrophinopathies, kind of the global term for Becker and Duchenne muscular dystrophy, and really target the damage and stop it where it's beginning, with muscle, with contraction-induced damage to the muscle. If we can stop that, then we can have a beneficial effect long-term. So with that, I want to thank you for your attention. And there's some contact information if you think of questions afterwards. I know I've seen questions pop up, but I can't talk and read and do all of that at the same time. So.
No, thank you, Joanne. Thank all three of you. That was a really great presentation. We did get a number of questions that came in. It might be easier for some of them if we kind of go back to the presentation to where they were asked, just so other people can kind of see some of the context. And some other ones that I had jotted down. So I think probably heading back to slide 15, and then we can kind of walk through there. There was an initial question, if you just wanted to clarify. Someone was asking if this was a steroid-based drug.
So it is not a steroid-based drug. It doesn't look anything like a steroid. And I would add that in this trial in Becker, we're looking at it without steroids. So we're treating patients with Becker that are not on steroids. And that would be an ultimate hope. Can the drug be used either with steroids or without steroids? So even in this early development, we're getting information on steroids or no steroids.
Great, thank you. And with this, there was a question that came in. So with the results from the ARCH study, this initial one with the Becker patients, you said you had some 6 month data. You're doing this biomarker collection. Are you collecting anything else in terms of functional measurements, imaging? Essentially, what other data is being collected, and for how long as well are you going to be collecting?
So in the ARCH study, we are collecting functional measures that hot off the presses. We actually showed on Friday, basically yesterday at World Muscle, we showed information on the North Star. and that those patients tended to improve. I mean, the study wasn't designed as a placebo-controlled study, but in the Becker patients who you would anticipate would be getting worse, got somewhat better. So that's encouraging. It's not a definitive answer, but it is something that we're going to be keeping an eye on. So we're looking at those kind of functional measures.
We also have patient-reported outcomes.
Mm-hmm.
I don't know if you want to talk about that.
Yeah, we are looking at things like pain, and we saw a trend towards improvement in the Becker patients. We are also looking with MRI in the phase 2 Becker study. So
Great, thank you. And we will as well. PPMD is going to have a small recap of some of the data that was shared at WMS. So we will capture some of the updates from Edgewise in that as well for the community. So you can keep an eye on that. If we go to the next slide. So there was just a question around the Canyon study, the ambulatory Becker and for adolescent and adults, just around with two, I guess. One, with the randomization is, can you talk about, is it 1-to-1, 2-to-1, placebo to active?
yeah, in the Becker study, it's 3-to-1.
3-to-1, okay, that's great. We like to see those numbers, so. The other question with that was around study sites, and I know that as you update on clinicaltrials.gov, that's when that will be made available. Are you kind of targeting similar sites, I guess, across the two studies with both Canyon and Lynx as you're kind of starting to roll those out?
Yeah, we are. There's a lot of overlap between the sites. Yes.
And I guess I do want to reiterate as well for those who were asking in terms of the sites, if you don't have one in your state, you did indicate there is some support to try and help those individuals get to sites where they can participate.
Yeah. We would cover all the expenses of travel to the site. Yes.
Thank you. If we go to slide 17. So a couple of ones here. First of all, so with this, the Duchenne study, about how many patients are you targeting for enrollment?
That's a good question. We didn't answer that question. It's 27 patients.
Okay, great, thank you. And Joanne, I know you did start to answer. There was a question around actually the use of why you needed to use steroids in the study, and I think you laid that out really nicely. And it, as it is standard of care as well. I guess I'd further ask with that because there were some other questions about, does this need to be used with steroids? Could it be with steroid naive patients? We had some questions as well looking at, are you measuring kind of fibrosis? So are you looking at inflammatory markers? Are you looking at over time how much fibrosis there is to see how much protection you're getting basically at the outset? So to prevent kind of that everything that triggers from missing dystrophin and how that kind of will affect the disease.
I'll turn that one to Alan to talk about biomarkers and also what the relevance of fibrosis in the animal models.
Yeah, and we're actually doing, we're really interested in steroids. So this first study, you have to be stabilized on steroids because that's the standard of care. Everyone tends to be put on steroids, so we don't really want to ask the question, does 5506 work without steroids? Because that's not really practical. Long term, well, that's a different matter. If we get great results, who knows? The landscape might change and you might be able to step off steroids, but this is a long way away, right? We are doing preclinical studies, so in mice, to look at combinations of steroids and 5506, and they work well together. And I think that's largely because steroids are a kind of immune modulators. They change inflammation, and what we do is protect the muscle that causes inflammation, right? So we're actually upstream from the steroids, and they seem to work nicely together. And actually, on the inflammation story, we actually do have some kind of fun data. In the Becker study, we just reported a couple of days ago, with 6 months of administration of 5506, what you're seeing with that kind of longer term data is really interesting evidence of biomarkers associated with inflammation changing slower. Right? So we've measured things like CK and troponin really quick because when you start taking the compound, those change quite quickly. But what we're doing now is what happens when you've taken the compound for a long time? We're actually seeing these signatures of inflammation changing. And as Joanne mentioned, that's absent any kind of steroid treatment. So we're upstream from steroids. We try and stop the damage at the very first step that leads to the inflammation. So as such, you should eventually affect all the fibrosis. And that's where these MRI measures come in, that we can image the fibrosis in the fat and muscle, and we can measure that changing over time, and hopefully we'll have a signal there.
And do that in a non-invasive way where you don't have to do muscle biopsies.
No biopsies. Yeah, I mean, when we put together the kind of what do we want from a medicine, our strong desire was to design a medicine that was applicable to everyone, that had low side effects, that was an oral medication, and that we didn't need to mess with people too much during the studies. And we're hopefully putting that into action over the next few years.
Great, thank you. And if we go to the next slide on some of the details of the study. So for the open label period, will all of the patients in that study be moved to the same dose, or will they just continue on the dose they had in the first 3-month treatment period?
Yeah, so they will continue on the same dose. As we learn more, then we may change that. But of course, we need to get the information, and the doses will be stepped up. So we'll start with the lowest dose, then move to a medium dose, and then a higher dose. So until we do all three, we can't know which one is the best.
Thank you. And if we go one more. So yeah, was very happy to see kind of the reduction in number of visits. I think that's always something that we talk about in terms of ability for families to participate in clinical trials is, having to take time off work, having to find care for the other, kids at home. So that's nice to see that it's 10 visits kind of over that 12 months. So is the frequency, is it higher at the beginning, or is it spread out throughout? And then
Exactly. So right at the beginning, when we are dosing the drug for the first time in kids, yes, it is. It's like it is more frequent. And then by the time we get to 6-9 months, it stretches out to every 3 months with telephone visits in the middle.
Great. And then on the next slide, you showed the device from side for monitoring activity. And this is something that I think may be new to some members of the community. Some of them may have participated in some studies that had limited use of this, but we haven't seen a lot. We've talked about it a lot, I think, in the drug development space.
Yeah.
Could you describe a little bit more about what the device is and kind of the expected wear time for patients if they're participating?
Yep. So the way that this works is that under the white part there's an adhesive. So you peel it off, put it on the arm, and then push the button. You just kind of push down, and then that deploys the tiny needle. And it is a very tiny needle. It's kind of like some of these glucose monitoring devices where they are now being used continuously with people. So it's a very small needle. And it stays there for about a couple of minutes. And as you can see, there's that little rectangle, that red rectangle. It removes a very small amount of blood. It is 100 microliters. So that is much less. It's drops of blood, basically. And that is, and basically you peel it off to allow those to dry, to the dried blood spots. Put it into a box, ship it back to the site. And so there'll be training at the first visit before we send you out with these, of course. And it will be training that is probably more articulate than what I just told you, I think.
Yeah, that helps. I think for that's and some people may be familiar with that of sometimes similar kind of pricks like that at the doctor's office. And then for the activity monitoring, is that every day? Is that for the entire
So,
length of the study that they have to wear that around
yeah,
the wrist or the ankle?
yeah, it will be for about 3 weeks at the beginning and then periodically during the study. So
Okay.
they take it off at night.
Great. And then if we can go to slide 21.
Yeah, I will say that early on, these monitoring devices tended to be these big clunky things. And so now it kind of looks like an Apple Watch. So, it can be cool.
Yeah.
So it's much better than it used to be.
That's good. You shared some really, I think, great things in terms of how Edgewise is considering clinical trials and trying to recognize that we do have a want in the community for broad ranges of ages and ambulatory status, and that was great to hear. Is using things like imaging, the ambulatory tracking devices, help go a long way. You also talked about combination therapies, and so just wanted to clarify again. You said that patients would be able to participate if they are in the Duchenne study, if they are on an exon skipping therapy.
Approved exon skipping therapies. So not in another investigational trial, but if they're on one of the 4 drugs that are available in the United States and they're stable on that for a year, because that's how long it takes to stabilize, then they would be eligible. So of course, yeah, and so we assume that there's going to be more therapies in the future. So we'll look to that as well.
And at the current time, would not be included if they were on a gene therapy delivering a microdystrophin, correct?
That's correct right now. Those are all investigational agents. So they would not be eligible at this point.
Okay. And then we had a few other questions come in as well. So following kind of that open label period for patients that are participating, will there be continued access for the drug at the close of that 12 month of the total study time?
Yeah, that is what we anticipate. When you are going to ask people to sign up at the beginning, to go to the regulators, to ethics, if you haven't got experience, it's hard to ask for a couple of years or 3 years, but our intent would be to keep people on and amend the study to do that.
Great, thank you. And then we had a couple questions about use with the drug. So is a question of around whether this could reverse muscle damage or just prevent muscle damage. They similarly asked if there had been any previous use of this drug outside of, I think, the trials that you've been looking at in terms of the healthy patients and the adults with Becker.
No, but I'll ask Alan about reversal.
I think just starting from the top, I think folks in the Duchenne community are quite often familiar with drugs that have been repurposed from something else. Oh, we tried it in inflammation and we're going to try it in this area. That's not the case with 5506. It was specifically designed for the kind of muscle weakness that occurs if you don't have dystrophin, or if dystrophin is not 100% functional. This is 100% for you guys. That's what it's made for. That's the goal, right? What was the other question?
So is it only to prevent damage, or was it able to?
Yeah. So
repair?
This is kind of a chicken and egg question in many ways, right? So think about if you're taking an ASO, okay, the best case scenario is you're going to now re-express a truncated dystrophin, and it will be, you'll essentially be a Becker person now, if that was 100%. Now, if you're active, your muscles are still getting injury. So you still have that relationship between I'm going to do something and then my muscles are going to get injured. And that really hampers the ability of muscle to positively adapt to exercise. As we all know, when we exercise, our muscles get healthier. But of course, that's a complicated relationship if your muscles are being injured every time you exercise. Now, the goal of this compound is to change that relationship. So you prevent the damage when you're active, but then that also allows your muscle to behave in a more healthy way and adapt to that exercise. So it actually gives you the potential for your muscle to improve, right? So it's going to prevent the damage. Does it reverse the damage? Well, if your muscle can now positively adapt to the exercise, well, in a way it kind of does, right? You're going to actually have healthier, more functional muscle.
Thank you for that explanation. That was really helpful. And then the other one that we had come in was around essentially continuation of the drug. So when looking at your animal models, do you need to stay on the drug to continue receiving benefit? Can someone come off the drug? And if they do, is that when damage would start again?
Correct. It's there, it's doing its thing. You come off, it's not doing its thing. Now this thing hangs around for a bit. So if you miss a couple of doses, no big deal. But remember, we're not changing dystrophin at all. So if you allow the compound to wash out, so if you didn't take it for a month, then essentially you're back where you started, and the injury will pick up again, right? That's the theory. So you kind of have to be on this thing for it to work.
In patients, if I'm remembering correctly from the start, it isn't daily oral tablet that the patients are taking.
Once a day. Yep. And they're little tiny pills, right? And we just, whether it's one or more than one to be determined. In the trial, it'll be more than one, but they're tiny. And
it's flavorless as well, so it's not really a yucky thing.
It's also usually helpful. With the Canyon study, so that, again, for those who are keeping track of the names, it's the ambulatory Becker study. Is that going to have some of the same kind of at-home monitoring pieces that we were seeing in the LINK study, which is the Duchenne trial?
Yeah, we do have activity monitoring in that study, and phone calls to check on people to minimize with stretching the intervals out there. We actually are not using the Tasso device in that study, though.
And then there was a question around anticipation, the dosing that you're going to use in the Duchenne patients. Is it going to be similar to what you've been using in the Becker patients?
Yeah, that's one of those hard things. You look at adults and you try to extrapolate to figure out based on height and weight what the right dose is in kids. And that's the reason for doing a trial like this, a 3-month trial, to see whether or not we have the right doses, because, in some things you take half the dose, or we just need to be able to adjust it. And so we'll understand more once we do that study and can make sure that we've got, as best we can, the right dose for a longer study for registration.
One of the benefits of going into Becker first is we do have a reasonably good idea of how much compound you need around for it to do what we think it's meant to do. So that really helps us in those dosage decisions.
Yeah.
Great. Well, I think that's most of the questions that have come in from the community. So thank all of you for taking the time to share that update with us. As your trials advance, as you get more data back in from the ARCH study, we of course hope you'll share that with us in the community. And we look forward to the next study getting off the ground and running. So thank you all.
Great.
Thank you very much.
Thank you, everybody.